Hi everyone. Today I'm going to talk about our research work on biosynthesis and characterization of deuterated microbial chitosan as part of my PhD research at North Carolina State University, and it was conducted at Oak Ridge National Laboratory. Chitosan is a remarkably versatile carbohydrate polymer, which is produced at industrial scale by chemical hydrolysis of the N-acetylated parent polymer, chitin. Chitin is abundantly found in structural components in crustacean shell and microorganism cell wall. Because some N-deacetylation occurs through a natural biological process, and because chemical N-acetylation can be in conflict, both chitin and chitosan are beta-1,4 linked copolymers of N-acetyl D-glucosamine and D-glucosamine, where the distinction between the polymers is empirically based on their solubility characteristics. Unlike chitin, which is insoluble in most solvents, chitosan has a higher than 50% degree of deacetylation and is soluble in diluted acids through the protonation of free amine group located at C2 position. Chitosan is a polycationic molecule. They do have some, in addition to the solubility and processability, it also has some attractive intrinsic properties and has been used in advanced material development, such as enzyme encapsulation or self-healing coating. In applications of using chitosan composite, it is very interesting to study the structure-function relationship using neutron scattering technique through characterization composite at solid states. In the example of using neutron scattering, it can be used to study structural features of entrapped enzymes at a solid state. The radius of duration of molecules can be obtained from scattering intensity with mathematical model. And this parameters is correlated to a difference between well-dispersed or aggregated molecules. Here's one example. The structure of GFP protein trapped in silica gel was studied using phase contract technique in a small angle neutron scattering experiments. However, when different carbohydrate polymers are used, such as cellulose, protein, together with chitosan, the scattering signal is from entire assembly, creating difficulties in differentiate biopolymers due to the similarity in the chemical composition. Therefore, we need to use deuterium to label one kind of the biomolecule in the assembly to apply the phase contract variation technique. Non-exchange duration of polysaccharides is challenging because of toxicity of deuterium to living organisms. To our best knowledge, chitosan deuteration has only been performed with yeast Pichia pastorase. Pichia has been used routinely to produce deuterated protein. In the work of chitosan deuteration, only FTIR spectrum was reported to demonstrate deuteration outcomes. No further quantitative data was provided about deuteration position and level. Also, the yield of chitosan from Pichia is relatively low. In our study, we report approaches to obtain chitosan from Pichia pastorase and other filamentous fungus, Rhizopus aureus. The free chitosan refers to chitosan that can be dissolved in 10% acetic acid with a polymer concentration of 50 milligrams per milliliter in our study. We first investigated cultivation conditions, chitosan extraction protocol, and the yields with these two microorganisms. The extractable chitosan from Pichia was about 1% of the dry cell weight, which is close to the value reported in the previous study. Growth in minimum media or rich media has limited impact of the free chitosan yield. However, because the rich media gives more higher biomass yield, the total yield of free chitosan was higher using the rich media the YPD. The FTIR spectrum of chitosan from 3 organisms elucidated a solubility profile of different samples. The relative extent of N-acetylation can be estimated from the IR spectrum by comparing the ratio of amide 1 bound at 1655 and that of the CH bond at 3450. This ratio in the Pichia chitosan is much larger than that of the chitosan from rhizopus and shrimp, indicating that the Pichia chitosan has fewer free amine group. The composition of the glucan can vary significantly in different fungal or microorganism species. We also observed the glucan structure in both microbial chitosan, which is not showing up in the shrimp chitosan. The ratio of band at 1640 to that of CH band at 3450 in the rhizopus chitosan is lower than the ratio in the Pichia, meaning the Pichia chitosan has higher glucan component. Therefore, both low degree of deacetylation and a high glucan component explained why Pichia chitosan has very limited solubility. The ability of chitosan to form a film is an important property for the future applications, such as the enzyme entrapment. So Pichia was not investigated further. The one function example is using chitosan for enzyme entrapment, where enzyme molecules are trapped inside the chitosan matrix, which can be stabilized to improve their longevity and reusability. The solubility of the rhizopus in 2% acetic acid and 100 millimolar sodium acetate is comparable to the conventional chitosan extracted from shrimp shell. And this allows the further processing of this fungal chitosan for protein entrapment. Therefore, we continued deuteration experiments with the rhizopus only. Without adaptation steps, rhizopus aureus growth was limited to 40% D2O concentration in a liquid media. Therefore, an adaptation process is required to grow rhizopus in higher D2O concentration. However, adapting rhizopus to 70% D2O liquid media took more than 1 month. In addition, in the submerged culture, the filamental fungus can grow into spherical pellets, and this will inhibit some substrate and product transport and lead to local toxicity and growth issues, and which will further drop the biomass yield in the cultivation. Most importantly, the biomass yield with the solid cultivation is higher compared to the liquid media with the same amount of carbon source, for example, the glucose. When cultivating rhizopus on the agar plate with the D2O from 20% to 70%, adaptation was not a requirement. The higher D2O concentration often lead to low cell density and earlier spirulation. Therefore, using the D2O as a deuterium source was not ideal for fungal chitosan deuteration, especially from the solid cultivation condition. Because only partial deuteration of chitosan is required for neutron scattering experiments, instead of using D2O as deuterium source, portioned glucose in the YPD media was substituted with a deuterated D7 glucose. We call it D-glucose. Surprisingly, the total biomass yield on the D-glucose plate was similar to that from the portioned glucose. Although the D-glucose substitution in the culture slows down the cell growth. The extractable free chitosan from the cell biomass harvested on D-glucose plate was about 83% that from the portioned glucose plate. Therefore, when we used a D-glucose as a deuterium resource with a solid cultivation, no adaptation is needed for rhizopus, and a similar biomass yield was achieved, and a comparable chitosan yield was achieved. The FTIR spectrum of portioned chitosan, H chitosan, the blue line here, and deuterated chitosan, D chitosan, the orange line here, dashed line here, from rhizopus are shown here. Deuterated chitosan from rhizopus has a unique peak between 2050, which is attributed to the CD stretching. In addition, the peak at 1154, corresponding to the carbon-hydrogen bending in H chitosan, was not observed in D chitosan. To further measure the deuteration level of the chitosan obtaining using our novel deuteration approach, we conduct portal NMR and the carbon-13 NMR on rhizopus H and D chitosan. The portal NMR spectrum of free chitosan samples are shown here. On left is the H chitosan, while the right is the D chitosan. From the portal NMR spectrum, the H3 to H6 atoms give characteristic signals between 3.6 and 3.9 ppm, and the H2 atom showed a single peak at 3.08 ppm. And both H and D chitosan show similar peaks at this position, but with much weaker intensity from the D chitosan. Further calculation of the deuteration level from the portal NMR suggests that almost half of the hydrogen position protons has been replaced by deuterium. However, because bias may be introduced during the sample preparation, for example, the moisture from the air, the obtained deuteration level may be different from their theoretical values. In addition, the peaks of H1 and amine and proton adjunct nitrogen were not observed from the spectrum, where they may overlap with the HDO peak at 4.79 ppm. So this overlap will lead to an inaccurate deuteration level calculation from the portal NMR. Therefore, we conduct carbon-13 NMR for further analysis. For the H chitosan on the left, 6 sharp signals at around 95, 74, 72, 68, and 57, and 53 ppm can be attributed to the polysaccharide structure. In a D chitosan spectrum, these 6 signals were also observed, indicating that chitosan was not per-deuterated. At the same time, deuteration also broadened the peaks. Extra peaks observed in the same region. An exception was C2, where no obvious additional carbon signal was observed close to the C2 peak, suggesting that deuteration degree at C2 is low. In order to separate the signal from C3, C4, C5, a carbon-13 attached proton test, an NMR study, was conducted on the D chitosan sample. C3, C4, C5 are all tertiary carbon atoms before deuteration. Therefore, their peaks can be integrated and separated from the CH to CD in the APT experiments. The carbon with deuterium gives negative signals, while the carbon with hydrogen gives positive peaks. Integration of these carbon peaks gives deuteration level on C3, C4, and C5, which they are highly deuterated. This is consistent with the pathway proposed in chitosan formation with glucose feeding. Carbon-13 NMR also shows lower deuteration level at a C2 position. The carbon connects to the free amine group. This is consistent with our proton NMR results. C2, the carbon atom connected to the amine group, might come from other nutritions according to some proposed pathway. Later on, a neutron scattering experiment was performed to characterize the structure of portiated and deuterated chitosan from rhizopus. The shape of the curves are similar, indicating overall polymer properties in solution were not affected by the substitution of deuterium. The continuously increasing intensity with decreasing wave vector Q in the Q range in certain Q range suggests that the structure of chitosan is consistent with long elongate particles. The feeding parameters for portiated and deuterated polymers are shown in the table. A significant difference in the mean fiber radius was observed for the processed and deuterated chitosan. Still, the maximum likelihood of fiber radius for the two chitosan form is similar. This means the radii of the individual fibers of portioned and deuterated have similar dimensions, but there is a broader distribution of fiber radius in the H chitosan sample. Therefore, deuterated sample can be useful as a model system to characterize the composite with contrast match experiments in neutron scattering. In this research, we obtained a novel deuterated chitosan with a higher molecular weight, higher degree of deacetylation, less impurity, and higher solubility with higher yield. We also observed positive outcomes of using fungal chitosan for enzyme entrapment. Deuterated chitosan also show potentials in characterizing the chitosan porting composite for enzyme entrapment application using neutron scattering techniques. Here, I will express my appreciation to my PhD advisors from Chair Dr. Sonia Selman, and also my committee and biochemistry advisor from Dr. Flora Mir from NC State Molecular and Structural Biochemistry. I also would like to appreciate the mentors and colleagues and scientists at Oak Range who helped me with this project and work together for data analysis and data collecting. Thank you.